Welcome, everybody, to this seminar covering the work of hyperacusis research. We advocate for people whose lives are destroyed, where ordinary sounds cause pain, discomfort, and distress. Our mission is to change that by funding research that uncovers what causes hyperacusis and drives the development of effective treatments. Since we were founded in 2011, our revenue and grants have steadily grown. To date, we've awarded nearly $400,000 to fund 10 research projects. Our expenses are minimal because we are staffed entirely by talented and hardworking volunteers. And I want to give a shout out to David Tawarji, who was a hardworking volunteer who put this webinar together. Our progress depends on advocates to fuel the research pipeline. We are in the middle of a campaign to get the word out about these opportunities. If you're here as a researcher eager to make a difference, consider applying for the next round of grants related to pain or loudness hyperacusis. Those who are studying tinnitus, which has a significant co-occurrence with hyperacusis, or at least with loudness hyperacusis, please keep this in mind in your work. And if you're someone living with hyperacusis or a loved one seeking to help, I encourage you to stay engaged, share your story, and if you can, volunteer to help us fulfill our mission. I'd like to introduce my friend, Dr. Stephen Barad, MD. He will take over the job of president of our organization on January 1st, 2026. Like me, he has a son who's suffering from a severe case of pain hyperacusis. In fact, one of the side benefits of this is that Lenny and Tom have gotten in touch with one another and are good friends as a result of this. So, Steve, take it away.
Thank you, Mike. It is, without saying, a privilege to step in to the best of my abilities into your shoes. Over the last 5 years, you've done an absolute outstanding job. And for those participating in this Zoom webinar, it's amazing where how far hyperacusis research has become over 12, 13 years ago. I mean, the fundraising all started off with a small golf tournament. We owe a lot to Brian Pollard, who was a founder, and it has grown rapidly over the last several years in terms of funding and, to me, more importantly, awareness of the issue. One of the common scientific meetings is what's called the ARO, Association of Research in Otolaryngology. And 5, 6 years ago, when I first attended because of my son's condition, I'd go around and the word hyperacusis, and in particular pain hyperacusis, was rarely, if ever, mentioned. And if you look at that as a keyword, that keyword has been mentioned with each successive year more and more and more. So one of our missions is to spread the awareness and increase awareness among all the participants and scientists in particular, for which we're really indebted for doing research along these lines. We have somewhat of a time frame of about an hour, hour and a half at most to get 3 beautiful talks in and a period for questions and answers. These are pre-submitted, and I picked out several of them for the sake of a time frame, so we just don't go too long on that. And hopefully we'll get those questions answered to everyone's satisfaction. And if not, I'm going to make a plug for a wonderful seminar that Dr. James Henry chairs. It's every Thursday, and you can get more information through our newsletter on how to participate and get many of your burning questions answered. We're going to start off with a 5-minute video from the BBC, a young lady who unfortunately is afflicted with pain hyperacusis. For many of you who may or may not be familiar with this condition, get an idea of what's happening with that. And then we're going to have a 10-minute talk by Dr. Henry, and then Megan Beers-Wood will follow that with a talk and she'll give you the subject, and then followed by that by Dr. Thanos from the University of Pittsburgh. Following this, we'll have probably about 15 minutes for questions. I'll ask each of the panelists, and maybe all three or individually. And then we'll try to wind it up with a family perspective. I'll give an account of what we've gone through as a family and what my son has gone through regarding this terrible condition. And then we'll have Michael wind it up. Okay. So again, I want to thank everybody for participating. We are truly grateful, and hopefully you'll find this webinar to be of interest. Our first speaker, and we are truly honored to have him, is Dr. James Henry. He is a distinguished audiologist. If you look at his website, he says he's retired from many years of practice in the field of audiology. But as an orthopedic surgeon who retired 2 years ago, I realized that you are never retired. And I'm sure that stands true for Dr. Henry. He is the principal or co-investigator for over 43 research grants. He has authored over 246 papers and publications. He has lectured on many topics, especially regarding tinnitus, all over the world. He has written several books on tinnitus-related subjects, and his most recent book on pain hyperacusis is a must-read for anyone interested. And you can get more information about this wonderful book from our website. I would go on and on with the many allocates, but again, as I previously said, we only have an hour and a half to talk. So, Dr. Henry, please take it away. We're really interested in what you have to say. Thank you.
Okay, well, thank you for those very kind words. I've got 10 minutes, so I've got to go fast. I'm going to give an overview, and I need to share my screen. And are we good? Does it look okay? Okay, and you can hear me. Okay, so what makes ears hurt? As Dr. Barad just mentioned, I spent about 6 months really digging into the literature after I retired. Tinnitus was really my area, but I was always interested in hyperacusis and misophonia. So I did a deep dive literature search to see what was in the literature concerning these conditions, and ended up writing this book. There's over 200 references. It's written like a scientific article, but it's also written in a way that anyone can understand. So I'm trying to make this information accessible to people. Also, this is a article that you might find interesting, and I can provide this. There's no charge for this article that I wrote along with Dr. Kelly John. And this is something I had to learn about. What is the difference between loudness hyperacusis and pain hyperacusis? So for starters, you know, if we're talking about what causes the ear to hurt, we first have to rule out otalgia. Otalgia is ear pain. It has nothing to do with exposure to sound. Loudness hyperacusis is a reaction to sound. Pain hyperacusis is a reaction to sound, but otalgia is just ear pain unassociated with sound. And we're not really here to talk about otalgia, although some people may think they have hyperacusis when they really have otalgia, and vice versa. So otalgia is ear pain mostly affecting one side. So it's unilateral. And there are primary causes and there are secondary causes. And we don't really have time to talk about that in detail, but just be aware that, you know, we always want to distinguish whether it's otalgia or whether it's one of these different forms of hyperacusis. And then on top of that, these different forms of hyperacusis are physical reactions to sound. It's the auditory system that's responding negatively to sound, versus there's two disorders that are emotional responses to sound. One is misophonia and the other is noise sensitivity. We're not going to talk about those today, because this is not ear pain. We're talking about what causes the ears to hurt. So misophonia is not the ears hurting, and noise sensitivity is not the ears hurting. Basically, misophonia is reactions to certain sounds, usually people eating, sniffling, body sounds, mouth and nose sounds for the most part. Noise sensitivity is this general emotional reactivity to sound. In general, sound is bothersome to these people. And then phonophobia is the fear. It's an excessive fear that sound will be uncomfortable or painful for any reason. So let's just focus on loudness hyperacusis and pain hyperacusis. So loudness hyperacusis is the experience of uncomfortable to unbearable physical sensations exclusive of piercing, burning, or stabbing pain in the ears and or the head when exposed to sound at intensity levels that are comfortable for most people. And most people feel like loudness hyperacusis is caused by increased central auditory gain. So think of gain as like a volume control in your brain, and it can be turned down or it can be turned up. And in general, for most people, the more sound you have coming into your ears, the more that volume control is turned down. And that's why sound therapy is often helpful for people with loudness hyperacusis, because it turns that volume control down and makes people less sensitive to sound. If we deprive ourselves of sound, then that volume control tends to get turned up, making us more sensitive to sound. And that's when sound can really be a problem, when that volume control is turned too far up. So that's what most people think is the usual cause of loudness hyperacusis. There's also the tensor tympani muscle in the middle ear, and that could be the cause of loudness hyperacusis. It could be both. And tensor tympani muscle is connected directly to your eardrum, and when it contracts, it stiffens the eardrum, and it's also part of your eustachian tube complex. So it's a complicated system, but if that tensor tympani muscle is spasming or working the way it's not supposed to, it can cause uncomfortable sensations in the ears. So if you feel like there's a balloon blowing up in your ear, or just pressure in response to sound, that could be the tensor tympani muscle reacting to sound. And the tensor tympani muscle is a very different system than the stapedius muscle. So we have two little muscles in the middle ear. The middle ear is the space behind the eardrum. It's the eardrum and the space behind the eardrum. And we have little bones that connect the eardrum to the cochlea, and that's how sound gets into the cochlea. And then we have these two muscles that can contract, and mostly the stapedius muscle is there to contract when sound reaches a certain level to protect the inner ear from sound that's damagingly loud. And it does a pretty good job of that normally. The tensor tympani muscle is a different circuit. And the tensor tympani muscle can contract just by thinking about sound being too loud. There's a lot of reasons the tensor tympani muscle can contract and be a problem. So shifting to pain hyperacusis, also referred to as noxacusis, it's the experience of burning, stabbing, or jabbing pain. That's what distinguishes it in the ears or the head when exposed to and or following any sound at an intensity level that would be comfortable for most people. I'm sure the other speakers are going to talk a little bit about this, but we have an auditory nerve that goes from the cochlea, the hair cells. You probably heard about the hair cells in the cochlea. That's what senses the vibrations and converts those vibrations to electric chemical signals that are sent through the auditory nerve into the brainstem. So there are type 1 neurons and there's type 2 neurons in the auditory nerve. The type 2 neurons connect to the outer hair cells, and the type 2 neurons, what's interesting about them is they can function as pain receptors. So if a person is feeling sharp jabbing pain in the ears in response to sound, it could be those type 2 neurons. There's only about 5% of them going through the auditory nerve from the cochlea. The other 95% are coming from the inner hair cells. So that's a very possible reason. This is all theoretical. We don't have clear answers on any of this, but we're getting ideas of what might be causing pain hyperacusis. And then back to the middle ear and the tensor tympani muscle, if we have myoclonus, which is uncontrollable twitching of the tensor tympani muscle, that could be the cause of pain hyperacusis. And there's also the central auditory pathways. Our auditory system can be hypersensitized in general. And let's see, where am I at on time? I probably have about 2 minutes. So I have all of these slides as a handout, and I'm going to send this to David when we're done, and he's going to send it out to everyone. So I don't have time to really go into detail on anything, but I want you to know this handout is available. There's the tensor tympani muscle connected to the eardrum. This is the tensor veli palatini muscle. So you can see how all of these structures are interconnected. The palate, the eustachian tube, the tensor tympani muscle, the tensor veli palatini muscle. So it's a very complex system, but this could be the source of ear pain. And then the middle ear, you've also got the trigeminal nerve, and that's a nerve that comes out of the brainstem and it innervates the face and the brain and the head and the ear. And so if there's problems with that trigeminal nerve, and things can inflame the trigeminal nerve and cause inflammation and other symptoms, the trigeminal nerve can be responsible for symptoms that people are experiencing. So there's another view of the trigeminal nerve. There's the auditory nerve, which is coming out of the cochlea, joining up with the vestibular nerve, and that's what goes into the brainstem. This is way too complicated to explain, but you've got the 3 rows of outer hair cells to the right, and then you've got the 1 row of inner hair cells to the left. And it's those outer hair cells that have the type 2 neurons going into the auditory nerve, and that's what we think could be the source of pain. The auditory pathways are obviously involved, and then there's also the possibility that pain hyperacusis and loudness hyperacusis are migraine related. And Dr. Hamid Jalilian is really the person who's discovered this and has followed up on it. But just be aware that we can have migraine symptoms without headache, and that would be called atypical migraine, and it's also referred to as central sensitization. Our whole brain can be hypersensitized to react to all kinds of stimuli, including auditory stimuli. And then this is an article that I can make available to people that really describes migraine and how it might be related to tinnitus and hyperacusis. And there you go. This information will be on my handout, and David will get that out to everybody. So thank you for the opportunity to give this quick overview.
Oh, wow. Dr. Henry, thank you for a wonderful introduction into some of the mechanisms anatomically and physiologically of the origins of hyperacusis. And before we get on to Dr. Beerswood talk, I want to share a video with you from the BBC. It was produced a few years ago and gives everyone an insight on what it is to live with this very painful condition. So David, go ahead and roll it, please.
Now, a mum of 2 has been telling us about her debilitating condition for which there is currently little or no treatment. Her name's Karen Cook. She lives in Southport, and Karen developed something called pain hyperacusis 18 months ago. It means that she is unable to tolerate most noises, and she's often forced to shut herself away from her family to live in silence. Well, Katie Walderman has been to meet her in Southport, but because of Karen's condition, we've boosted the sound levels. But you might wonder, this is the explanation for why some of Katie's report has had to be whispered.
Sound is everywhere. You know, it's like air, you can't escape it.
For the last 18 months, this has pretty much been Karen's life. Alone, in silence and in pain. Shut away from her young family, watching the world go by from her window.
My house is a prison. Sound keeps me prisoner.
Everyday sounds like the wind in the trees, passing traffic, or even her children playing causes Karen crippling pain.
Something as beautiful as my children's laughter and hearing their voices is torture for me.
18 months ago, Karen's life turned upside down when she developed a debilitating hearing condition called hyperacusis. There are different types, but hers means sound causes excruciating pain and is accompanied by tinnitus.
It's like somebody poured burning lava into my ears. And my head burns, pain all over my head, pressure behind my eyes, like a migraine type pain, but all through my face. Pressure, like you just want to sort of split your face open and release the pressure. So I'm in constant pain, constant pain.
But Karen hasn't been able to find any treatment that eases the pain. She wears earplugs and defenders even when she's at home alone. Her only way to communicate through whispers.
Just tell us what impact this has had on your life. Devastating. Devastating. I miss being a mum. I miss the clatter and the noise when they come in from school. I miss just life. I sat and watched them through the window, opening their Christmas presents. Because it was too loud for me to be in the room with them. And they'd come up to the window and show me. It's completely erased me.
What's been the hardest part for you?
It's losing my best friend. Happy birthday to you. Life is an adventure. We just go out. No plans. We just go out for the day. Wherever we ended up, fantastic. Travelling of any description. We bought a caravan during COVID, hiking, taking the boys to football matches. Any excuse to get out and about, that was us. Everything that we knew as a family has changed.
The NHS says reintroducing sounds like white noise can help build back tolerance for some with the condition, but for others like Karen, there's very little that provides relief.
For pain hyperacusis, there really is no treatment available. I mean, for me, time, quiet environments, and really just avoiding loud noise is the key to managing it and not making it worse.
The pain and mental torment of isolation is taking its toll.
Honestly, if I didn't have children, I'd have given up. But we're going to try and find someone, anyone who might be able to help. I'll try anything and everything before I give up.
Katie Walderman there speaking to Karen Cook.
I hope that gives many of the participants in today's webinar a bit of an insight in the lives of someone with hyperacusis, pain hyperacusis in particular. My son Lenny has a very similar lifestyle, unfortunately. Our next speaker is Dr. Megan Beers-Wood. She received her doctorate at Emory University in immunology and molecular pathology, her postdoctoral work at Johns Hopkins School of Medicine. And during that tenure, she received a grant funded by Hyperacusis research in 2022 and through 2023. The grant basically shed light on the mechanisms and potential treatments for pain hyperacusis. And I may make a side note, again, a lot of this incredible research cannot actually happen without generous funding from all those that are concerned. And so we make a plea for that. Megan Beerswood is currently now an associate professor at Vanderbilt University School of Medicine, where she heads the Harmonic Laboratory dedicated to study NIHL, studying noise-induced hearing loss, excuse me, and the association of hearing loss with pain hyperacusis. And personally, we are really indebted for her valuable membership in our scientific board. And we are, Megan, very grateful for your participation in tonight's webinar. So Megan, what will your topic be today?
I am going to be giving a short presentation entitled Modeling Auditory Pain. I'm going to go over, I hope you can see this, I'm going to go over some of the things that I've done in the past and kind of where I'm planning to take this in the future, just briefly. But hopefully I won't take up too much of your time. Thank you for those kind words. I am very happy to be showing you some of the fruit of the grant that I received from Hyperacusis Research in this presentation. So going back to type 2 afferents, I'm going to do a brief background on what they are. This is a diagram looking down at the sensory epithelium that we use to hear in the cochlea. It's kind of shaped like a snail. And here the blue dots are the inner and outer hair cells. The inner hair cells are on the medial side, and the outer hair cells are these 3 rows out here. In gray and black here in the middle, these are the cell bodies of all of the neurons that help you hear, and they're in the spiral ganglion. There are 2 types of spiral ganglion neurons. There's type 1 spiral ganglion neurons, and those are the ones that contact the inner hair cells and really send the information to the brain about what you hear, both frequency and intensity. And then there are these secondary types of spiral ganglion neurons, type 2 neurons, that are only about 5% of the total number of neurons, and they have a very unique morphology. They send their dendrite out to the outer hair cell, make this 90-degree turn towards the base of the cochlea, and then they contact up to 16 outer hair cells. And we know a lot about the type 1 neurons, but we really don't know a lot about what the type 2 neurons are and what their role is. But these 2 groups of neurons are sending afferent signals to the brain. And afferent just means that they're collecting information from the ear and sending it to the brain. So we started in my previous lab to try to understand what do type 2 afferents do. And so one of the first things was a series of papers looking just at the type 2 afferents. What do they look like? And some of this is also historical. One of the things we know is that they're unmyelinated, which means they lack a sheath around the neuron, which you can think of like insulation. It usually makes neurons send signals faster, but these do not have them. Also, we knew that through a series of studies that they share gene expression such as tyrosine hydroxylase, the gene for CGRP, and the gene for substance P with these other neurons called C fibers. And C fibers are the pain sensing neurons that are located in places like your skin. So then we try to look more into what do these neurons respond to? And that is what led to these studies, some before my time and some during my time, where we found out that the type 2 afferent fibers could actually respond to hair cell damage and to ATP. And ATP is interesting because the only reason that ATP would ever be released is if a cell was damaged, because suddenly the ATP that's normally inside the cell would be released, and that can cause activation of neurons. And in general, this would mean that type 2 afferent neurons can respond to tissue damage, which again is something that pain sensing neurons would usually do because they report damage and tell your body that something's wrong. Then we did a series of studies. We're trying to understand if type 2 afferents change after noise exposure. There's a lot known about how type 1 afferents change after noise exposure, but not much is known about type 2s. And so there was a small study that we published in 2021 where we showed that in normal mice, you can see here at the bottom of these outer hair cells, you see these ribbons, which are the connections between the outer hair cell and the type 2 afferent neurons. And you can see that in this cell, there are none, in this cell, there's one, and in this cell, there's only one. And that was kind of a typical pattern. However, after noise exposure, you could get an increase in the number of these ribbon synapses. Here in these cells now, there's more red dots at the base of the outer hair cells. And this was a very interesting observation because it meant that instead of, for instance, losing synapses, there after noise exposure, you might actually be getting an increase in input into these type 2 afferent neurons. Then we went on to try to understand that was only looking at pictures, right? So it wasn't saying anything about the actual function of the neuron. So then we went on to do this study where we put a calcium sensor into the type 2 afferent neurons. And that means that the type 2 afferent neurons will fluoresce when they're activated. And we looked at adult cochleas and tried to damage the cells around a type 2 afferent and see if it would respond. So to see if it would respond to tissue damage. This was extremely difficult to do in adult mice. However, if you noise expose the mice, we could then get the type 2 afferents to respond. And so this meant that there might be some sort of sensitization that was occurring with noise exposure. And so here I have a picture, I have a video. In this red circle, there will be damage to the epithelium. You can see this early fluorescent wave that goes out through the supporting cells. And then after a little bit of a delay, you'll start to see a very bright fluorescent line, essentially, that is the activation of a type 2 afferent neuron. So all of this says there are some similarities in type 2 afferents with the way they look and the genes that they express with pain sensing neurons. And it seems like they are changing, it's potential that they could change their activity after noise exposure. But there's one major question still remaining, which is, are these changes in type 2 afferents associated with pain? So one of the next things that we needed to do was characterize auditory pain in normal hearing mice to be able to even study this question. So first, I just want to, as a transition, make the point that everyone can experience auditory pain. And that is when a sound is really loud, it can be painful. And this has been documented in the literature over and over again. So a normal hearing adult human can experience pain to sound. But then we need a way to transition from humans that can tell us that they're in pain to laboratory animals, which cannot tell us they're in pain. And so the way we did this is we adapted the facial action coding system, which is a way to tell whether a nonverbal human is in pain. And there are stereotyped changes to the face that occur. This had been done previously in laboratory animals, many different types of animals, including mice. And it was called the mouse grimace scale. However, there were a few things that were not ideal about the mouse grimace scale. So we actually adapted it based on another researcher's work, so that we put a camera facing a mouse that was mostly in profile, and then taught a deep neural network to find 13 dots on the face of animals. And this ended up coming up with 6 different measurements of the face that we could track frame by frame of video. And we added all of these measurements together and correlated them to a baseline to generate a facial grimace score when the animal was listening to different levels of sound. The other way that pain is often characterized in lab animals is by looking at their movement or changes to their movement. And so we also made a new score where we looked at the body position of the animal just by tracking one dot of the nose tip around the chamber. We were able to capture these measurements that roughly correlated to rearing, turning, exploration of the cage, and whether or not the animal was hunched. And again, just like with the facial grimace measurements, we added all of these measurements together to form a body position score. And so this is a very busy slide, but the main thing to say is that what we found is that if a mouse was listening to over 100 decibels of sound, it had a significant its behavior was significantly changed from baseline. And this was true for both the facial grimace and the body position. And so here I have some examples to show you of a tracked mouse in no sound, which you can see is acting fairly normally, exploring the chamber, and moving around. This is an animal listening to 70 decibels of sound. And then still exploring the chamber, very much so. And then this is an animal listening to 110 decibels of sound, and this is broadband sound. And these two graphs are pretty busy, but one of those reasons is because there are these two threshold lines. Those two threshold lines were created based on a known pain model that we compared this to. And so we can say that these three groups of mice listening to 100, 110, and 120 met or exceeded that threshold of pain. We went one step further. This, by the way, this study is now on bioRxiv and will be submitted very soon. We went a step further and looked at a mouse that has no ability to hear sound. Everything should have developed normally, but its cochlea lacks the ability to send signals to the brain. And this mouse showed no significant changes in any sound level, either in its facial grimace score or its body position score. And here is some video of one of those mice listening to 110 decibel sound. And so what we concluded is that you need a functional cochlea. A normal hearing animal needs a functional cochlea to experience auditory pain. This does not mean that there isn't involvement from the middle ear, because there is a feedback loop from detecting sound, sending those signals to the brain, and then having feedback both to the cochlea itself and to the middle ear, as we were just learning. So there can still be other parts of the auditory pathway that are involved, but but clearly detection of sound is important for the generation of auditory pain. And so with that, I just want to say briefly that my future plans are to set up a model of loudness hyperacusis, which we already know exists, using noise exposure. We can do this with either a temporary threshold shift noise exposure or chronic noise exposure, and then do our analysis of behavior in these animals and assess the number of outer hair cell ribbon synapses and type 2 dendrite number underneath the outer hair cells, and see if there's any correlation between increased number of ribbons and the behavior of the animals, to really try to start linking pain behavior to the type 2 afferents or not. But I think that is a really critical next step to understand how type 2 afferents may be involved in this pathway. And so with that, I just want to thank everyone that I've worked with on this, especially we affectionately call it Team Grimace, Ben Sekel and Amelie Vallis, or Vias, and Anna Kohler, and then and my previous mentors, and then all of our different funding sources. Thank you.
Megan, thank you. That Dr. Beardwood, that's just a wonderful talk, great overview, and we look forward to hearing more from you in the very near future. Thank you. Our next speaker is Dr. Thanos Tzounopoulos. He is quite accomplished. He is the director of the Pittsburgh Hearing Research Center. He is endowed professor and vice chair of basic research in the Department of Otolaryngology at the University of Pittsburgh School of Medicine. He received recently the Bellucci Pioneer and Research Award held at the annual Bellucci Symposium in Omaha. And it was during that symposium that I had the pleasure of meeting his absolutely wonderful family. Dr. Tzounopoulos has done extensive work also on noise-induced hearing loss and tinnitus, with the exciting development of a potential therapy that could mitigate against this from happening. This research, along with the help of Dr. Manoj Kumar, was the basis for our Hyperacusis Research Grant 2023-2024. And again, to all those that are here attending this webinar, another plea for donations. Without money, none of this can happen. And again, it is my honor to introduce him, and we are I may speak for the entire board. We are truly indebted to for his participation. Thank you, Dr. Tzounopoulos. Please give us what you're going to talk about, and we look forward to hearing from you.
Sure. You can see me, hear me, right?
Yes, perfect.
That's good. First, thank you very much for the very kind introduction. And thank you and the Hyperacusis research for inviting me here to talk about some of my research. As Steve said, this work that I'm going to discuss with you today, I don't have slides, but I would like to discuss a few things. Is a collaboration between myself and Dr. Manoj Kumar. He's an assistant professor at the Pittsburgh Hearing Research Center, and we've been working together on this. So what I'm going to say is more relevant for loudness hyperacusis, as you're going to see. And it was great introduction by Jim Henry to talk about loudness hyperacusis. And if you recall, he said that one of the major mechanisms associated with loudness hyperacusis, central gain increase, right? So what is this? We try to understand a little bit more what is this and what are the mechanisms underlying these central gain changes. So, okay, we're using animal model, we're using mice where we expose them to loud sound. So we give them noise-induced hearing loss. And when mice are exposed to loud sound, all the structures that you saw in the cochlea, they suffer some damage. And the end result is that the input that goes to the brain from the sound is getting reduced, right? So less amount of nerve fibers, less input goes to the brain. So the brain notices that and is trying to compensate. It knows that not enough sound comes into the brain anymore in terms of signal, and the brain tries to increase that knob, that gain that Jim Henry was talking about. This is a good thing, you know, because although there is a peripheral damage and less input comes in, with the remaining and smaller input that comes in, the brain can still perceive the sound, right? And it can alleviate some of the symptoms of complete deafness. But then these neurons in the brain, they compensate their activity, so they have no. But in a lot of times, what we see even an overcompensation of the activity. Like it reaches the input in response to sound reaches the control levels before the hearing loss, but then in several occasions it becomes more actually than the control. Looking at the positive symptom, right? There is more sound if the knob is too high up, if you will. So this change has been associated both with tinnitus and hyperacusis as a result of noise-induced hearing loss. So what we tried to understand several years ago is what makes these neurons more active, right? Although they receive less input, what makes them fire more, right? Because that's the activity of neurons. They fire action potential, right? That's a currency. They are current. And to make the long story short, after many years of research, we found that these neurons, they have a molecule that's called, it's a potassium channel that is not working well. But what is potassium channel and why do we care? So potassium channels are these membrane proteins that allow for potassium ions to go in and out of the cell. And basically when these channels open, because they can be closed or open, these channels, potassium comes out of the cell and basically it's a major break. These potassium channels are a major break of activity, right? So when the cell wants to quiet down, it opens the potassium channels and the activity quiets down. So it turns out that these neurons, these central neurons, these potassium channels don't open when they're supposed to open. And then this break now doesn't exist. And then that leads to more activity, which, as I can tell you, it can be beneficial because it allows us to hear a threshold, but it can also be maladaptive, if you will. And it can, when things are not getting well adjusted, it can lead to tinnitus and or hyperacusis. Okay, we found this, and that was a specific potassium channel called KCNQ. Then at the time, there was a drug outside in the market, an FDA-approved drug called retigabin, and that was used for epilepsy. And then this drug would keep this channel open, right? It would have the opening of the channel. But right as we're having these findings, there were some negative symptoms, symptoms associated with this drug, side effects of this drug, and people developed blue skin coloration and retina. The retina would become blue. So that was bad news, and the drug was removed from the market. So then, but then when we used this drug, we're able in animal models, and even before it was removed, to correct both the neural hyperactivity, but also we're able to correct the perceptual correlates of this hyperactivity in animal models of tinnitus and now hyperacusis. So when we collaborated with the medicinal chemist, and he created a more specific molecule, small molecule, that can open these channels, but that can also alleviate the side effects of these channels, right? And that's the RL81. So when we use, after we noise exposed, when we give mice RL81, we see that we can quiet down both the neurons, but also the perceptual consequences of this quiet down, that mice don't show anymore hypersensitivity to sound, perceptual hypersensitivity to sound, and or tinnitus. So that is the story, basically, that we have a small molecule that can keep this channel open and then can help the nervous system to quiet down its response to sound. And then in itself, that triggers another plasticity in the brain. Because as I told you, the lack of sound tells the brain compensates for that. But then when we use this drug, we found out that it has long-lasting effects because it causes a corrective plasticity that it tells basically the brain to tone it down. Right now, we're trying to understand how long is the beneficial effect, for how long the beneficial effect lasts of this drug. We're also doing all this that I told you is preclinical, right? This is all in animal models. And we try to look at the toxicity and the metabolism of this drug. And then also we have a second lead candidate because there were some toxic effects. I think they're manageable. And the other thing that I would like to tell you, there are a couple of companies out there which we've been talking about, and these companies, they also generate openers for these channels. They're using them for epilepsy. Their clinical trials are for epilepsy and for depression. We've been talking to them, and if their development goes faster than ours, we'll be happy to pair with them and share our expertise to have clinical trials both on tinnitus and hyperacusis. So I would like to stop here, and thanks again for listening.
Thank you. Thank you very much, Dr. Tzounopoulos. A great overview. Typically, we were thinking of giving you a family perspective, but I think before that, I would like to start a little bit of a Q&A, and beginning with you, Thanos, if you don't mind. You mentioned this with RL81 that trials are about to begin, or have they begun yet?
Okay, they have not begun yet. We're still in the preclinical development, and basically what we're trying to address is the safety of the compound to make sure that there's not any toxic effects. This is advancing. As I told you, this has been there has been some findings that have also led us to create a second lead candidate that could alleviate some of the toxic effects that we found. And so this is where we're at. So we're still in the preclinical development of these compounds.
For the audience, let's say things work out with your research in regards to, well, there isn't much of a way of side effects and various things like that, and you get an FDA approval to begin a trial, phase 1.
Right.
How are you going to get the participants in that trial? I mean, will
Right.
we be able to help you, or how are you going to go about? Do you have any idea how you're going to go about that?
So, because so far what we have thought more about was about tinnitus, right? And now, of course, we're thinking about hyperacusis. And I collaborate with an excellent clinician scientist. His name is Greg Basura he's at University of Pittsburgh, who has also experienced clinical trials with tinnitus. So perhaps we would start with tinnitus first, because in the clinic we get many patients, many tinnitus sufferers, right? So I don't think it would be very hard, at least for tinnitus, to have people get them interested to participate in the clinical trial. As a matter of fact, I get several emails already asking me about that, but also in the clinic, Greg Basura gets a lot of interest. Regarding hyperacusis, if we reach that state, obviously, I guess maybe we're close to that state, talking to you and also go to the clinic and talk to Greg, how many patients we have that suffer from hyperacusis. But for tinnitus, there is a lot of demand.
Well, one of the things we all know is that there's a significant crossover with tinnitus and hyperacusis.
Absolutely.
Particularly significant, painful tinnitus, in a sense, devastating tinnitus where people are homebound, and similarly with pain hyperacusis. And I hear the figures are somewhere upwards to 80%. So perhaps when you begin these trials, and you get these participants, there could be a questionnaire or validity scale as to the association of hyperacusis symptoms. And on a side note, look at that to speed that along for us.
That's right. Absolutely. Absolutely, we would go for that. And as I said, I'll talk to Greg, as you know, I'm not so much into clinical, I don't see patients. Mostly I'm in the lab, right? But yeah, I'm talking to Greg, and I will let him know, and that's a great idea. And we will do that.
Good. We appreciate that. I have a question for Dr. Beerswood. And these come from questions that have already been pre-submitted. Are the type 2 pain fibers related in any way to the middle ear contractions? Are these pain fibers independent, and or could they possibly activate the trigeminal pathway, which is one of the theories for pain and facial pain?
Right. So I'm not totally sure about the trigeminal pathway. It's one of the things that has not been done is a full mapping of where the type 2 afferent fibers, who they connect to in the brainstem and beyond. However, they do seem to connect to an area in the brainstem that has some cross-modal plasticity. So there are some connections between potentially the type 2 neurons and other senses. But I do know that there is definitely, if the type 2 afferents are feeding into the brain, then there are signals being sent back out of the brain that will include changes to the middle ear, especially the stapedius muscle, as we were talking about. So there is a potential feedback loop that could be partially not controlled, but that the type 2 afferents could be a part of. So I'm not saying that they're mutually exclusive, but I think it's really interesting to continue to look at how each of those parts may be involved in something like pain hyperacusis.
Okay, thank you. Another question for you, and maybe Thanos could chime in. And I think I asked you this about a year ago at the Iowa conference. The models for the CGRP alpha antagonists, I think they were given to prescribing recommendations for humans. And again, for everyone, these are drugs that are used to treat migraines. So that's one way of administering things. I was wondering, and I've seen this at a poster, what does everyone think about direct administration to the middle ear and inner ear through a tube through the tympanic membrane, much like tubes that are administered quite often in infants for otitis? Do you think that has some potential benefit? And if it did, if there was a way of keeping that tube in, would that be a more direct effect on the neurons, for example, as far as inflammation?
So I think there's a lot of research going on best ways to deliver different drugs to the middle and inner ear. I would say that, for instance, there are different hydrogels and things that have been placed at the round window to allow things like even steroids over time to kind of diffuse into the inner ear. And then, of course, one of the ways of giving gene therapy into the inner ear space is also would be through the tympanic membrane, or actually with it retracted and then injected into the inner ear. And that's the nice thing about the ear, is that you essentially have a controlled space to put a drug. Obviously, there's always a problem if you just inject in the middle ear space about things coming down the eustachian tube. But it's definitely under a lot of that's just an entire area. Drug delivery is a huge topic in the inner ear and middle ear space, including even using nanoparticles to push magnetic nanoparticles to push drug through the round window and into the inner ear. So yeah, I mean, I think that one thing that we always have to think about with hyperacusis patients is whether they would tolerate that sort of approach for a drug, and whether or not it would be better to give something systemic, or I guess on the offhand, could you mitigate some of the systemic side effects if you were able to do a procedure in the middle ear? But obviously, you want to make sure that the patient is comfortable and is willing to undergo any sort of intervention. I don't know if anybody else has any
And a
thoughts.
good study would be to see what is the the amount of drug delivery via different mechanism rather than sub-Q or something, or sub-Q being subcutaneous injection versus something orally. So I believe that's certainly very promising. Thank you. Jim, I have a question for you, if you don't mind. This was brought up in some of the pre-questions, and something I've often thought about, and certainly certainly consistent with my son's issues with pain hyperacusis. The question being, what can unilateral hyperacusis patients teach us about the possible underlying pathology of hyperacusis? In other words, why does it seem to affect one ear more than the other? Is there any insights you could give us as to what might cause that, and how people, in the auditory brainstem, how things are affected by that?
That's a really interesting question. I would say one thing we know is that anything that can damage the ears can cause hearing loss, hyperacusis, both types of hyperacusis, and tinnitus. And so some people have asymmetric or unilateral hearing loss, same thing with tinnitus, and same thing with these different forms of hyperacusis. So you'd have to look at what is causing the damage and what is causing the symptoms. And from my experience, most people who have loudness hyperacusis, and I don't know that many people with pain hyperacusis, the experience is bilateral. They experience it in both ears. So, but we do know a lot of people have unilateral tinnitus, asymmetric tinnitus, and the same thing can happen with hyperacusis. It's just a matter of what caused it and what's the underlying mechanism. Other than that, I don't know. I mean, it would really take a person, that would be a research project in itself.
That's interesting. As far as mechanisms, and perhaps you can answer to this, and this is not a question I'd ask you, and everyone can chime in. Hyperacusis seemingly and constantly is associated with some type of a noise-induced traumatic event. But I happen to know, and I've heard on several occasions of people having hyperacusis-like symptoms from a head injury. And does that tell us a little bit about maybe damage to the auditory cortex? And as Dr. Tzounopoulos noted, with the higher centers being hyperexcitable, perhaps that injury higher up in the brain, the cortex, can lead down the chain to the auditory center, to the auditory centers in the midbrain, and give you those symptoms. Does that shed any light? Does anybody have any thoughts about that? And this is for all three panelists.
I'll let Megan and Thanos respond. They'll know more than I do. But one thing about a traumatic brain injury is it can affect all aspects of the auditory system, from the middle ear all the way up to the auditory cortex. So noise exposure damages the hair cells. We know that. And that
Right.
causes further issues on up the auditory system. But a traumatic brain injury can affect the entire auditory system. So I'll let the other two respond.
So that this is
Make any-
another area that's like very actively under investigation. And I would say there in the research world, there's definitely people break down between blast associated mild traumatic brain injury versus something like a traumatic brain injury where your head is hit, like a concussion. But Jim is totally right. We don't quite know. It can affect every single step. So we're not sure how each one of those components is exactly involved. One of the reasons why we've kind of developed this pain assay in my lab is because we're interested in looking at some of those models and trying to understand whether any of the hearing loss or changes to auditory perception are associated with pain afterwards, as opposed to just associated with changes in hearing.
Okay, thank you. Thanos, any
Yeah,
ideas?
I have not much to add. I agree with both of them. And yeah, the traumatic brain injury affects the whole auditory system, plus it affects non-auditory systems, right? Like systems that control pain and emotion. And so all these things can come together and create such a disorder, right? But it's auditory and all the other systems that affect.
Okay, thank you. Many of the sufferers in the pain hyperacusis community in particular cannot go out and cannot work. And I'll talk a little bit about this with our personal situation. And one of the things often encountered is there really isn't an understanding by people that assign claims examiners, things like that. Well, how do you really know you have hyperacusis? What's the documentation of this? And much like someone with diabetes, severe diabetes, for example, the biomarker might be elevated glucose or what is called an A1C or something like this. And so I was wondering, are there any thoughts, and Megan, I'll ask you this first, research into specific biomarkers for hyperacusis, something that could validate a particular person's complaints in regards to obtaining, for example, disability benefits?
Excellent question. So I think this is actually one space Dan Polly and his group have been working on this, and so is Kelly John, is thinking about ways to track responses to sound and whether those can be tracked to hyperacusis. And so there was some recent work showing that even just movements of the face could be changed in different sort of disorders of hearing, including misophonia, tinnitus, and hyperacusis. And those are some things that I think would be you could have a baseline recording and then have periodic like retesting essentially to show that your responses to sound are altered. And so that's kind of a different way of thinking about a biomarker instead of a blood test.
Okay.
And although I recognize that that does include sound, which could be troubling. So obviously we are going to be continuing to think about ways to look at something like a biomarker. One of the problems is going to be sampling. Is it going to be enough to get something out of a blood test? Or would you need to sample from the middle ear and inner ear space, which would be much more invasive to really be able to tell? But ultimately, we would love to be able to follow someone's progress also over time, either through something that's not invasive, if possible. And also through something like recording of the face or pupil dilation, be able to do that at levels of sound that do not cause pain, but still cause some behavioral changes.
And you see that as a documentation for
Yep.
someone to bring to somebody. Look, this is validated research
Yep.
in Dr. Bearswood's lab, and
Mhm.
this is gospel, and this is verifies that I have hyperacusis, for
Yeah,
example.
well, I mean, obviously there would have to be clinicians involved, but I think that's the idea, is developing some of these more behavioral based tests so that they could be implemented in the clinic and there could be documentation that there are changes that show a difference from a typical hearing
Okay.
adult or a person.
Okay, got it. Great. My son Lenny introduced me to something that I've been using it, but previously I've really never heard of it. It's called ChatGPT. And now I use it for everything. And I still see people in a clinical setting and they ask me questions and I go, let's get another opinion from a doctor. And they go, well, where? There isn't another one here. And I go, no, I'm asking my phone. And it's been incredibly helpful. In that light, what are your thoughts, Thanos and Megan, about the use of artificial intelligence as far as acceleration of findings and research? Is it something you're now employing in your lab, or do you see a real benefit for this? Thanos, maybe?
Yeah.
Yeah.
I mean, this is not something that I'm currently using in the lab. I can tell you that I have not. I don't know. I mean, of course, artificial intelligence, I haven't thought deeply about it, but artificial intelligence, of course, it can help. Like sometimes we use it even, it can help writing even or getting information. But personally, I haven't used it yet to a big extent in the lab. So perhaps Megan might have some more
Yeah, I was thinking in terms, not to interrupt,
Well,
findings of research. Well, this can lead to this, to this, to this, and maybe get some ideas other than your basic inert knowledge.
yeah, so I'm actually really excited about this in certain ways. So what we've been doing is, so one of the ways to look at the face for a while would have been to try to hand place dots on the face of every single frame of our video, which is 46,000 frames for a 25-minute video. And when we tried that for even a 2-minute video, that took 3 hours. So by using a deep neural network and training it to place the dots where we wanted it to, it massively gave us a huge amount of data out and reduced our amount of time that we had to take to annotate video. We're continuing to use machine learning tools to analyze other things about our video. For instance, like being able to pick up on more sophisticated behaviors that the animals might be performing. And as I mentioned, there are many 3D facial meshes that are available on the market to be able to use these machine learning tools to analyze faces, for instance. And one thing that I'm really excited about at Vanderbilt is that there's a data science institute that can help us transform some of these large data sets and try to learn more from them. So like now, we have the videos. What can machine learning tell us about something about the video that we weren't even capturing? And so there's a way to really kind of accelerate what we can do, even with a small number of people. And so I'm really excited about that usage of AI in research.
That's terrific. Well, the talks and the questions and answers really quite illustrative. And again, we appreciate everyone's involvement with this. I want to speak a few minutes about our family, and in particular my youngest son, Lenny. His injury was pretty much noise induced from playing music too loud. He was a bass guitarist and just could not play music loud enough. Went to a lot of concerts. We knew nothing about protecting your ears at that time. It's like, why bother? You won't get the full effect of the entire experience. But something developed with him, and he started having a distortion of sound, not knowing what was going on. And we took him to various therapists. We tried everything, and he saw a physician in Arizona, tried the sound therapy to no avail, did his best with that over a period of a year, and just came to a point where he's been living with it for over 11, 12 years, if not longer. And this has certainly affected his life. And much like we saw on the BBC video, he's basically imprisoned by his house. There's concern about going outside, unanticipated noises, things like that. He's ventured around quite a bit, and fortunately, he has a wonderful person named Kirsty Tatterson, who's basically his other better half, and so he's got company, and they do a lot of things together, and we're certainly appreciative of that. One of the biggest things, and I'm sure that would be something encountered by people with families who have this condition going on is, at least initially, a lack of empathy regarding this person's issue. What do you mean you have tolerance to sounds? I don't. Is this something you're making up? What's happening? To Lenny's condition, he was in high school, one of the more popular kids. He had every weekend, there's, groups of 20, 30, all getting together. Many of them were his bandmates. Just a wonderful situation. I was in high school, had a great experience, played some baseball, had a lot of friends, but nothing to what he had. He had a large group. He was very funny and still is, has a great sense of humor. But one by one, as this condition became more manifest, he started losing all his friendships. And basically, he only has one friend. And this is quite sad in this community because you can't break out. And to that effect, I cannot thank enough our technology. So there's such a thing as a computer, and he can network with many other people and realize that he is not alone in this. So he has formed many virtual friendships, which are certainly better than being without. So again, as incoming president of Hyperacusis Research, I just want to thank everybody for their involvement, particularly with our three esteemed scientists. Excuse me, and make a plea for donations. Research cannot be done without funding. So $1, $5, $10, even more, we'd appreciate anything anybody wants to give, and certainly move this all along. And to that, I give way to our president, Mike Maholchic, for closing comments.
Steve, I want to thank you for your closing comments because they basically tracked very well with my closing comments, so I don't need to repeat myself. But I want to thank you for moderating this discussion. I want to thank David Troorgi for setting up the technical aspects of it, and especially our presenters, Thanos, Megan, Jim. We are counting on you guys to change our lives. It's terrible. My son's got it, just like Lenny, and has had it for 12 years. And you've got no life. And I think if you have no life and hope, you're way ahead of someone who has no hope and no life. This fall, your generosity can only go twice as far with a $50,000 matching gift challenge. This will help us fund you guys, and it will bring relief, understanding, and hope to thousands living with the pain of sound. We want to keep you up to date with our progress by sharing our newsletter, and that'll include links to a monthly discussion forum hosted by Dr. Jim Henry. So thanks for showing up tonight on this. You can watch it later on if you missed it. And let's beat hyperacusis. Good night.
Good night, everybody, and thank you very much for participating. We really appreciate it. Thank you.
Okay.
Okay.